Hello, friends and well-wishers. This is going to be the first stab at going through my quiz review. So we'll see how far I get, and if this is taking more than 15 minutes, I'll probably break it up into two. Okay, so this is what we need to know for the quiz today. Okay, first of all, you need to know something about brain regions and their functions. In broad strokes here, the cerebral cortex, which is the outer bark of your brain, is responsible for higher functions. Let's see if I can list a few off the top of my head. I don't know, your personality, your sense of identity, your ability to reason abstractly, a lot of your sensory perception, motor functions, integrating motor functions. Well, wait, I might be getting a bit ahead of myself here. We really just want to think about higher brain functions. Anyway, basal ganglia and the limbic system are responsible for movement, motivation, and memory. So memory is tied into limbic system. Movement, basal ganglia, and these motivations, the 4 F's, and yes, they did say this in lecture. Fight or flight, feeding, and fucking. Somewhat regrettably that he had to say that in lecture is kind of weird, but it's true. Okay, brainstem nuclei. Their function is to modulate higher brain functions, and they do so via the variety of neurotransmitters that are produced in these regions and that project to other areas of your body, in particular higher brain areas. And the medulla is all about regulating your vitals, so breathing, respiration, blood pressure. Okay. So this is from Wikipedia. I'm just going to read through it. The main components of the basal ganglia are the striatum, which is the caudate nucleus, and the putamen, the globus pallidus, the substantia nigra, nucleus accumbens, and the subthalamic nucleus. So there's a lot going on in the basal ganglia. The largest component, the striatum, receives input from many brain areas, but sends output only to other components of the basal ganglia. The pallidum receives input from the striatum and sends inhibitory output to a number of motor-related areas. The substantia nigra is the source of the striatal input of the neurotransmitter dopamine, which plays an important role in basal ganglia function. The subthalamic nucleus receives input mainly from the striatum and cerebral cortex and projects to the globus pallidus. Each of these areas has a complex internal anatomical and neurochemical organization. Okay, so for the neurotransmitters, the real thing that we need to know is the source of it, or the sources of the neurotransmitter, and a little bit about it. So the dopamine sources would be the substantia nigra. The substantia nigra is a brain structure located in the midbrain that plays an important role in reward, addiction, and movement. So dopamine, substantia nigra, midbrain. Substantia nigra is blah, 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 blah. It appears dark because there's, melanin? Yeah, high levels of melanin in the dopaminergic neurons. Parkinson's disease is characterized by the death of dopaminergic neurons in the substantia nigra, in particular the pars compacta. Although the substantia nigra appears as a continuous band in brain sections, anatomical studies have found that it actually consists of 2 parts with very different connections and functions, pars compacta and pars reticulata. The pars compacta serves mainly as an input to the basal ganglia circuit, supplying the striatum with dopamine. The pars reticulata, on the other hand, serves mainly as an output, conveying signals from basal ganglia to numerous other brain structures. So that's one source of dopamine. The other source is the ventral tegmental area, or the VTA. It's a group of neurons located close to the midline on the floor of the midbrain. So dopamine, VTA, midbrain. Dopamine, substantia nigra, midbrain. Okay. Dopamine, midbrain, 2 sources. VTA and substantia nigra. VTA is the origin of dopaminergic cell bodies of the mesocorticolimbic dopamine system, and so it's widely implicated with in the drug and natural reward circuitry of the brain. It's important in cognition, motivation, drug addiction, intense emotions related to love, and several psychiatric disorders. The neurons project to numerous areas of the brain from the prefrontal cortex to the caudal brainstem and several regions in between. Okay, so we did dopamine, now we got to do serotonin. The source is the raphe nuclei of the brainstem. So this is sort of a diffuse collection of nuclei that string along the entire brainstem, so along the midbrain, pons, and medulla. That's all I say about that, but we know there's probably going to be questions about SSRIs and mood and depression and all these things. Okay, so norepinephrine. Source: the locus coeruleus of the pons. Help if I can always include of the pons when I say locus coeruleus so that I can implicate norepinephrine with that specific place. Norepinephrine from the locus coeruleus of the pons. Okay, so there's going to be a question about implicating norepinephrine with, like, general alertness. So the way that works is that norepinephrine is going to bind beta-2 receptors in the brain, in the cortex, and it's going to lead to a decrease in activity of the calcium-activated potassium channels. Now, calcium-activated potassium channels are themselves inhibitors. Okay? So if we bind beta-2, decrease these calcium-activated potassium channels, we will decrease inhibition, which, the double negative, this means that we're going to increase cortical neuron stimulation, and this is why we have greater alertness when we're on or having accelerated dosages of norepinephrine, or increased secretion. Okay, so we've done, just to review, we did dopamine, substantia nigra and VTA of the midbrain. We did serotonin, the raphe nuclei of the brain stem, scattered throughout, norepinephrine, locus coeruleus of the pons. Now we've got oxytocin, which is from the paraventricular nucleus of the hypothalamus. Okay, so they want you to know what oxytocin does. Well, it inhibits neurons in the amygdala. So the amygdala is like your fear area, and it's also associated with social olfaction processing. So basically, when you get an oxytocin burst, you generally let your guard down in terms of stimuli that you would consider frightening or smelling bad, I guess. So, that's implicated in pair bonding, owing to trust and empathy that it engenders. This is an example of bottom-up influence of neurotransmitters. Okay, so there's oxytocin. Next we need to talk about so this must be about enkephalins. Where do I actually have it? Okay, I'm going to just go down here and summarize what's happening. So this is all about inhibiting pain signals from reaching your brain. So basically, the periaqueductal gray matter, which is gray matter surrounding the aqueduct between the third and fourth ventricles, this guy's going to release enkephalins. So this is where the neuronal cell bodies are, but the neurons project to the raphe nuclei. So we've got enkephalins causing serotonin release in the raphe nuclei, but the raphe nuclei axons are projecting to, or there might be some intermediate connection, I'm not exactly sure, but basically this hits the substantia gelatinosa. This substance, this gray matter, is found in the posterior horns of the spinal cord. Okay, so you've got stimulation of the substantia gelatinosa by these serotonergic or these serotonin-releasing axons. Okay, so what happens then? Now you are going to stimulate the release of enkephalins from the I don't know if there's interneurons there, but basically, okay, you've got substantia gelatinosa. These guys are going to project to and synapse with first-order neurons. So this is your incoming pain signals from your, say, like your hand if you're getting burned. And so normally, you know, your first-order neurons would bring your pain signals, your alpha or your C pain fibers, like I don't know, whatever. Those guys are incoming, and normally they would have to like synapse with interneurons in the spinal cord. They'd cross over and go up the spinal thalamic tract. But what happens is these enkephalins that are being released are going to bind to receptors on the first order neurons that are going to inhibit the release of substance P, and so that signal is never gonna cross, or it's gonna be inhibited. So that's an example of top-down pain modulation, because something coming from your brain, initially from the periaqueductal gray matter, is influencing pain signals coming back up the spinal cord, so it's top-down. Okay, then we get into axon growth cones. Okay, so what do you need for actin? Oh, maybe before I go on, incoming C and A delta fibers. That's what I missed. Right, so, you know, I wouldn't be surprised if we got asked about orexin, which, if my memory serves me, the source is, oh, I didn't actually say the source for oxytocin. Oh, paraventricular nucleus of hypothalamus. So it's stored in the posterior pituitary. It's like a posterior pituitary hormone, but it's it actually, what's that thing? The pituitary hypothalamic axis, or like portal circulation there, I guess is how it gets there. Right, orexin, so that has to do with like hunger and wakefulness cycles, and I have another video on that I can go cross-reference, but I should include orexin in the study session because we talked about it. Okay, so what do you need for a growth cone to get growing? So this is like you've got, you know, a neuron cell body, and there's like an axon that's starting to come off of it. It's budding and it needs to like get somewhere if it's going to transfer information. So what do you need there? So you've got actin treadmilling happening. This is inside of the axon growth area. So this involves filamentous actin polymerizing, depolymerizing to globular actin, so F-actin and G-actin. You need some kind of connection to the extracellular substrate to anchor the actin filaments. And once you have that, you've got myosin, which is interacting with the microtubule to propel it forward. So basically you need an anchor, you need actin treadmilling, and you need myosin to anchor to the anchor and yank the growth cone along, and that's how you can get it projecting or propelling forwards. Okay, sensory neurons from sensory, okay, this is just a statement of fact that I need to know, is that sensory neurons growing from sensory cells in the PNS traveling towards the CNS make use of guidepost cells. So if you're thinking about peripheral nervous system towards central nervous system, sensory neurons, you want to be thinking guidepost cells. Okay, for commissural neurons in the spinal cord. Okay, so there's going to be both homo and heterophilic interactions happening here. Okay. There's a bunch of different proteins, and so I made just like this little point-by-point way of thinking about them. So you've got your axon extending from a neuron, from a soma in the central nervous system. Now, initially, there's going to be attraction of the DCC, which is the deleted colorectal cancer receptor. Like, well, the gene that codes for it is this DCC. It's also called the receptor itself, we're just going to call it DCC. On the growing axon, it's attracted to the ventral midline by diffusible protein gradient of netrin-1, which is released by the floor plate cells. Okay? Crossing of the midline is facilitated by the interaction of tag1 on the axon with NR-CAM from the floor plate. Okay, so at the beginning we have, so these are both attractions. DCC wants to come get netrin-1 as we approach the floor plate. Tag1 wants to interact with NR-CAM. Okay, as the axon crosses the floor plate, there's this interaction that results in floor plate no longer presenting tag. Ooh, no. There's an interaction with the floor plate that results in axon no longer presenting tag 1. Instead, it comes up with robo. Okay, so robo interacts with slit in the floor plate that is responsible for short-term repulsion that causes the axon to take a turn. And then slit and netrin together facilitate long-range repulsion. So let's see if I can do it without looking at the computer screen. Long-range attraction is DCC and netrin-1. Short-range attraction is tag1 and slit. No. It's tag1 and NR-CAM. Robo and slit is short-range repulsion, and then netrin and slit is long-range repulsion. DCC netrin-1, tag1 NR-CAM, robo and slit netrin. Okay. Also, extracellular matrix adhesion molecules, for example, laminin and fibronectin, provide a favorable substrate for neurite extension. And so these ECM proteins can be associated with the axon or with like whatever it's crawling along, like basal lamina or whatever. Okay, synaptogenesis requires the assembly of pre and post synaptic specializations. So for the neuromuscular junction, you've got agrin on the neuron interacting with muscle-specific kinase on the myocyte. And basically this results in an aggregation of acetylcholine receptors using this rapsin scaffolding. And that's taking place on the postsynaptic element. Now the myocyte is going to be releasing laminin B2, which is to changes in the axon associated with substructure development, so the vesicles and protein scaffolding. Okay, in the central nervous system, you've got the same sort of story, but you've got neurexin from the axon interacting with neuroligin from the postsynaptic. Sorry. So neurexin and neuroligin shake hands, and so neurexin is going to induce your postsynaptic changes, and your postsynaptic neuroligin is going to induce presynaptic changes. So they shake hands. Okay, in development, one muscle fiber will have many axons innervating it, but these polyneuronal innervations must be pruned so that each fiber has only one branch innervating it. And this is mediated by axonal competition, the details of which I'm not very clear on, but okay. So you can have three states of a voltage-gated sodium channel. There's like two gates. There's an activation gate and an inactivation gate. Now at rest, the channel is closed. The activation gate is closed, but the inactivation gate is open. When the channel is activated, everything's open. And when the channel is in a deactivated state, the channel is closed. The inactivation gate probably ends. Everything's closed. Okay. So, for sodium, potassium, and calcium voltage activated channels, these are big things that we have to know. So each of these channels is made up of 4 alpha subunits. So like you've got like big alpha subunits. Okay. And each of these subunits has 6 domains, S1 through 6. So the pore is formed by the so-called P-loop between S5 and S6. So you've got 4 of those coming together and making the actual pore. Now the S4 domain, the S4 helix, is the voltage sensor in all of this. So it has a number of positive residues which are usually exposed to the interior of the cell, indicating that the channel is closed. Now they sense when it's time to open, and they swing their positive residues to the extracellular space, causing like rotations at S5 and S6. And that's what opens the channel. Right, because we established that the P-loop is between S5 and S6, and so this conformational change in S5 and S6 causes the pore to open. And also, so now you've got these positive charges sort of swinging to the outside, and these are needed to strip the hydration from the ion, because basically a sodium ion, for instance, it's got this positive charge. It's going to be sort of attracting 4 water molecules to it, and these need to get like stripped away in order for the ion to pass through the channel. So the positive charges on the S4 helix are going to, I believe, enable that to happen. You also want to know that the intracellular beta subunits are involved in inactivation of the channel. Okay, so I talked about the activation gate and also the inactivation gate. Well, this is like that beta subunit of the channel is involved in inactivation. Okay, so patch clamp configurations. First of all, patch clamp techniques are all about measuring voltage through a single channel and seeing how a single channel behaves. And though you can take whole cell recordings, so I don't want to be too limited in that. But so this is how you want to think about this. You take your micropipette and you're going to touch it to the cell membrane, and the micropipette is extremely small in diameter, like one micron. So you can essentially isolate like a single channel or very few channels like within your patch, and you're just gonna like put light suction on it. So that forms a giga-ohm seal, and this is called the cell-attached configuration of the pipette. So at that point, you could just withdraw the pipette and sort of bring with it that patch of membrane. So now you've got a situation where the inside of the channel is actually facing the outside environment relative to where it was in the cell. And the outside environment of the outer face of the channel is now facing the inside of the pipette. So this is called an inside-out configuration. Now, if instead of before pulling, if you applied enough suction to rupture the cell, then you're getting a continuous passage between the pipette and the inside of the cell. and this is where you can do your whole cell recordings. But we can go further. We can withdraw the pipette once we've broken the cell membrane, and somehow, I have yet to see, I've scoured YouTube and looked extensively to get an idea of how this actually happens. I'm not really sure. But somehow, after breaking the cell membrane, if you withdraw the pipette, you get a configuration where the outside face of the channel is actually still facing the outside solution, or the bath, and the inside is now facing your micropipette. So you've essentially swapped inside environments from being inside your cell to being inside your pipette. And this is called the outside-out configuration. Okay, the last two things here we have to reason about the kind of measurements that you would make doing these patch clamps. Is that what they're called? Oh, John. Yeah, these are patch clamp configurations. Like, what kind of measurements could you get? So, okay, if the pipette solution and the bath solution are in equilibrium, there should be no current measured through the channel. Now imagine you apply current. In this case, unless specified, applying current means applying negative to the inside, because the electrode is actually inside the pipette. Okay, so we're going to apply negative to this patch clamp configuration that was in equilibrium. So what happens if the channel's open? Okay, well you would get negative current. You'd get current running out of the cell. Okay, so if you're going to be measuring your I guess your potential against time as this happens, you're going to see negative deflections. At the same time, if you applied a positive current when the channel is open, you get a positive deflection. So this is not exactly rocket science here. Okay, now how do we think about the shifting? Okay, so basically the take home there is that if you are applying 0 voltage, it's kind of hard to think about. If you're applying no charge to the inside, and the inside and outside are in equilibrium, when the channel's open, you get no current flowing, or no net current flowing. Now, suppose that there was not chemical equilibrium between the inside and the outside of in your experiment. So we'll say the inside of the pipette versus the outside of the pipette. Suppose you were holding the inside or the inside had a concentration of potassium resulting in, say, what do I want to say, 90 millivolts, and the outside has a concentration resulting in 5 millivolts. Okay, so clearly, assuming that this is an outside-out configuration, potassium wants to get out, because that's the way that the channels would be oriented, and that's the way that are the channels symmetrical? No, they're not, because we've got, anyway, I'm not going to get too bogged down in this. Basically, you've got this plus 85 that wants to, get out, right? So when channels open, you're going to have potassium rushing out, and so you're going to see positive deflections when the thing opens. Okay, so the question in the last thing we had no current flowing when we applied no current, but now we've got this disequilibrium. We've got to make up for 85 positive. So if we apply 85 negative current, then you would expect when the channel opens, you're still going to have equilibrium. if we applied any sort of positive current, well, this is just going to accelerate potassium rushing out, and so we're going to get positive deflections. And in fact, even negative currents up to negative 85 will cause outward current. Any negative current under minus 85 would result in an influx or negative deflections when the channel's open. Okay, that's about all I got here. Thank you.